Hello, and we're glad you've joined us for this live webinar, When Cell Growth is Critical, Evaluating CO₂ Incubator Parameters for Cell Therapy. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. And it's brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit them at www.thermofisher.com. So let's get started. You can pose your questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience any technical problems seeing or hearing this presentation, just click on the Support tab found at the top right of the presentation window or report your problem by typing it into the Ask a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process of obtaining your credits. I now present today's speaker, Mary Kay Bates, the Senior Global Cell Culture Scientist at Thermo Fisher Scientific. Welcome, Mary Kay. You may now begin your presentation.
Thanks, Susie, and hello, everyone. Thank you for attending, and thank you to everyone at LabRoots for presenting this event. Part of my job with Thermo Fisher is to work with our R&D engineers to communicate what is needed for cell and gene therapy applications to maximize ideal conditions for cell growth in our cell culture incubators. As part of this work, we have compared different engineering approaches for the control of the CO₂ incubator, especially looking at recovery following a door opening. And we looked at recovery also. And we looked at uniformity. And to be honest, we were surprised at the variation that we found. So the basis of my presentation today is to share some of these findings because just comparing a list of functional specifications does not necessarily indicate how the incubator performs in practice. Also, incubators have been used in cell culture since the 1950s, and I think that many of us have sort of forgotten how each parameter affects the cells themselves. And so we will also consider how variation in these separate incubator parameters can affect cells. I'm excited to share this information with you, partly because I wish I had understood this better for applications in my own previous work in cancer and gene therapy, and also because this information has been helpful for some cell therapy institutes directly, so I wanted to share it on a broader basis in hopes you will find it helpful also. So let's get started. I've set some goals for today's presentation. At the conclusion of my talk, I hope that you will understand how each parameter controlled by the CO₂ incubator affects cell growth. You will discover how incubator design differences can affect the performance of the incubator and thus the health of your cells. And finally, I hope that you will learn that just like in reading a journal article, you should be able to critically assess equipment features based on the data provided in brochures and technical information from the manufacturer. So it's not just about comparing a list of functional specifications, but you're also looking at practical performance, and you can ask for these data. So that information should guide your choice because ultimately you have to perform IQ, OQ, PQ testing as you implement this equipment into your processes, and you don't want any surprises. So as by way of introduction, let's kind of take a step back and look at cells that we're culturing. So as we've trended towards cell therapy and gene therapy applications, we're seeing use of and growth of more sensitive cells. So many of you are growing immortalized cell lines like HEK293 cells, for example, just for production of cells or from products from cells. But also many of you may be growing stem cells for direct use in human therapeutic applications. Certainly, we're seeing a lot of advancement and a lot of expansion in looking at growth of immune cells. And finally, human primary cells. If you think about autologous cell therapy, where you're taking cells from the patient, modifying them, and then putting them back in the patient to treat a condition in that patient. So the thing about cultured cells, of course, is they're constantly sampling the milieu around them that in which they are. So they're looking at temperature, humidity, atmosphere, gas concentration, nutrients and stimulants and repressors in the growth medium. Companions, are they alone in that cell culture, or are they touching other related cells that may trigger different gene expression or differentiation? And they're responding to those conditions. And since the CO₂ incubator is there to mimic the in vivo conditions, or the conditions in the body from where the cells originated, that means that each parameter affects cell growth. And as we've just established, cells respond to changing conditions. So the take home there is that variation in your incubation conditions can cause variation in your process results. Because as your cells respond to different conditions, that can affect gene expression or protein expression and so on. So now I want to take a look at each of the parameters provided by the incubator individually. What are their effects on cells, and what happens when we see variation in the incubator, and then compare different technologies for achieving those parameters. So I think with an incubator, the first thing you think of is temperature. So let's begin with temperature. So incubator temperature is there to mimic the body temperature. So the reason we culture cells, especially human-derived cells, at 37 degrees is because that's body temperature. And we know that cells are extremely sensitive to temperature. And if you think about part of your immune response is to develop a fever, and you know that you don't feel very well when your temperature, your body temperature, is above normal 37 degrees Celsius. We also know that cells are generally okay for a few days at refrigeration. So lower temperatures are generally okay, and this is also physiological. When you wake up first thing in the morning before you've had that first cup of coffee, your body temperature is often lower than 37 degrees. It might be 36.5 degrees Celsius. But higher temperatures are detrimental to cells. In fact, cells cannot withstand more than a few hours of 2 degrees above normal. And of course, this is in culture, not outside the body, where a fever is also detrimental, but maybe not as detrimental as cells kind of naked in culture. So with that in mind, let's look at how recovery after a door opening of temperature can vary depending on the design of the incubator. So what I'm showing here is a table showing recovery to 37 degrees, or 98% of 37 degrees, after a 30-second door opening. So the inner and outer door of the incubator was open for 30 seconds. And this is a common test for functional performance of an incubator. So left to right, the small blue bar shows recovery in 5 minutes after a 30-second door opening, and this is an incubator with a circulating fan and a temperature probe located in the incubator chamber. Going left to right, the yellow bar shows recovery from a 30-second door opening to 37 degrees Celsius in an incubator without a circulating fan. And you can see that recovery is a little bit slower. If you put the temperature sensor in what's called a bypass location, and I'm going to talk about bypass sensors later in the talk, but essentially it's outside the incubator chamber, that slows the recovery even further, so that's more than 10 minutes to recover to 37 degrees after a 30-second door opening. And then the last bar shows you more than a 30-minute recovery time for an incubator without a circulating fan and with a bypass temperature probe. So my point here is that even though incubators generally look very similar from the outside. Different technologies, so different design and engineering that goes into those incubators can produce very different conditions for cells. The other thing that you might see in a brochure, for example, is that in trying to hurry or speed the recovery after a door opening of temperature, you can see an overshoot of the desired temperature. So here's a recovery graph from one incubator on the left that shows a speedy recovery to about 36 degrees, and then it slows as it approaches 37 degrees. And then if you focus on the graph for incubator B, you can see it's actually a slower recovery, and then when it hits 37 degrees, it keeps going, so it overshoots 37 degrees. And as we've just established, higher temperatures are actually more detrimental for cells. So overshooting that set point of 37 degrees does not produce faster recovery, and it can be detrimental to cells. So I wanted to provide some tips to maintain temperature in your current incubator. So I assume that not everybody on the call today is going to run out and buy new incubators based on my talk, but what can you do in your current incubator to maintain temperature better for better growth of your cells? So number one, and you're going to hear this again, limit the number of times that you open the incubator door and limit the duration of the door opening. So for example, if you go to the incubator and you pull out a stack of cells and you carry them over to the work surface and then you go back and close the incubator door, that's not ideal. So is there a place next to the incubator where you can put the cells and then quickly close the incubator door to better maintain that temperature? Another tip is to preheat the growth media, and I hope that everyone is doing this, to preheat your growth media to 37 degrees, partly because, of course, putting cold or room temperature growth media will shock the cells, very bad for them. But also preheating that growth media helps better maintain the temperature in the incubator. If you want to, you can also preheat the water that you provide in the humidity reservoir. That will also help maintain the incubator temperature because it won't have to work so hard to heat up that cold water that you put in. This is one we see especially in newer buildings where at night when no one is in the laboratory, the ambient temperature of the laboratory will change in order to save energy in the heating and cooling system. So that affects how the incubator functions, especially in warmer climates. If the laboratory heats up overnight, that can really negatively affect how the incubator is heating the cells inside. Often, even in clean rooms, I walk in and I see building air ducts for heating and cooling for the room in the ceiling or on the wall, and they're right pointed on the incubator. So those, of course, blowing intermittent cold or hot air on the incubator, again, will change what the incubator is experiencing and how it has to heat the cells. So one thing to do is you can use kind of a passive air duct diverter to change that airflow so that it's not blowing directly on the incubator. I don't think this happens very often, but sometimes we all love to see sunlight coming into our workspace. So make sure that if you do have windows such that sun is shining on the incubator, use some window blinds just to keep that sunlight from shining on the incubator. And then finally, if overheating is a concern for your incubator, an incubator with a backup temperature probe helps prevent overheating in the event of a failure of that primary temperature sensor. Okay, so that's temperature. Now I'm going to look at the second most important parameter provided by the CO₂ incubator, and that is gas concentration. So I'm going to start with a little bit of biology because I think sometimes we think about the incubator as just a place to put the cells and then we can get on with the next step in our day. And I think it's important to take a step back and understand the biology behind all of these parameters and how they affect the cells. So why do we provide carbon dioxide? Well, I think everybody knows that the reason we provide carbon dioxide in an incubator is to balance the pH. And so this is actually mimicking what's going on in the bloodstream. So in this case, the pink kind of cloud represents my sort of cartoon of the lobe of a lung, and the arrow represents a blood vessel. So the purple cells are blood cells that are deprived of oxygen. They're bringing waste carbon dioxide, which is a waste byproduct of every cell in the body. And so they're actually less red as they're carrying carbon dioxide instead of oxygen. And it turns out that the oxygen concentration in the lungs, where it's kind of full of waste carbon dioxide produced by the cells, is 5% CO2, or 40 millimeters of mercury pressure. And of course, that's the most common CO2 setting on the incubator is 5%. So the incubator is mimicking what's going on in the body. We also have oxygen in the lungs, of course. Oxygen in the Earth's atmosphere, the air we breathe, is between 20 and 21%. But in the lungs, the oxygen concentration is much lower. It's 14%. And this is because oxygen doesn't diffuse very well. So, of course, what's happening in the bloodstream in the lungs is that waste metabolism, waste CO2 from the cells is being expelled into the lungs, and you exhale it in your breath, and oxygen is coming in. Now, the other thing that's happening in the bloodstream, along with the CO2, is there are natural bicarbonate ions, which serve as a natural buffer in the bloodstream. And the bicarbonate ion concentration is 24 millimolar, which is equal to 2 milligrams per milliliter sodium bicarbonate, which is the buffer most commonly used in cell culture growth media, and the same concentration in Eagle's minimal essential medium, upon which most growth media are based. So as you can see, the conditions we're providing in the incubator are there to mimic exactly what's going on in the body. Now, we can look at it from a biochemistry level, but in short, the growth media include that buffer, the sodium bicarbonate, to work with the CO2 and balance the pH. And it turns out that unlike oxygen, carbon dioxide gas dissolves and diffuses really easily in liquids. So there's an equilibrium with the gas phase, or that provided by the incubator, of the carbon dioxide and the carbon dioxide in the growth medium. And so the sodium bicarbonate is there to give off and take up ions, or to balance and buffer that pH. Now, it turns out that fluctuations in the carbon dioxide can affect the cell morphology and growth, and I think most of us working in cell culture are aware of this. But my point here is not just about the incubator, but also when you're looking at your cells under the microscope, take an extra minute or two to look at that cell morphology. Are the cell membranes nice and tight against the dish if you're working with adherent cells? Do you see any vacuoles in the cytoplasm that might be an early indication of toxicity, for example? And that might be something to look at for your carbon dioxide. So now let's think about how do we control carbon dioxide in the incubator? So there are a couple of different sensor technologies. An earlier technology is called thermal conductivity, and in this type of sensor, the gas is not directly measured, but it comes as a function of the relative humidity and the temperature, and that's in accordance with Boyle's gas law, that the pressure of the gas will change with other gases in the system, including water vapor, and also with temperature. Many of us are using infrared sensors. These are great for connecting with monitoring systems, for example. Infrared sensors were first introduced in incubators in 1984. And an infrared sensor is essentially like a light bulb. Like an older incandescent light bulb, it has a tungsten filament that, when electrified, gives off light. And so the gas enters the sensor. It interacts with the light, all wavelengths except 4.3 micrometers, at which CO2 gas absorbs light are filtered out, and then that amount of CO2 gas triggers an influx of carbon dioxide gas as needed. Now, the thing about a tungsten infrared bulb is that tungsten filament degrades over time as it's heated, and so because of that, the light intensity decreases over time as well. When the light intensity decreases, that contributes to decreased accuracy of the measurement and drift of that measurement. So one of the options is to get a newer technology where instead of using tungsten, a silicon light source is used to provide that light. And this turns out to be much more stable and much more long-lived than tungsten bulbs. It's kind of the equivalent of moving from an incandescent light bulb to an LED light bulb. And so as I said, a silicon light source is much more stable and long-lived than an older tungsten-style infrared light bulb. So how does this affect the recovery after a door opening? So just like with temperature, we're going to look at how these different technologies affect the practical function of the incubator. So in this graph, we're looking at time to recovery to 5% carbon dioxide in minutes following a 30-second door opening of these different incubators. And again, these charts, these measurements are based on some of our internal testing, but also taken from brochures from different manufacturers. So again, going left to right, the silicon-based IR sensor, infrared sensor, will recover from a 30-second door opening in about 4.5 minutes. A newer technology thermal conductivity sensor, which has relative humidity compensation so that measurement is no longer dependent on humidity, also recovers quite fast. The green bar indicates a bypass infrared sensor, which is a little bit slower to recover, and this makes sense because this sensor is outside the incubator chamber, and again, we'll talk about that a little bit later in the talk. If you have an incubator with an IR sensor, but it does not have a circulating fan to speed recovery, you can see that doubles the recovery time. That's the yellow bar. And then finally, if you have an older style thermoconductivity sensor and it's outside the incubator chamber, so it's a bypass sensor, I've actually had to truncate the graph there. So that recovery is 40 minutes from a 30-second door opening. So again, I hope I've demonstrated that different technologies can produce very different conditions, and of course that's going to affect how your cells grow. So one final note on gas and atmosphere in the incubator, and that's, I wanted to point out that many of us are also using hypoxia in an incubator. The point of this is to decrease the oxygen concentration to mimic what's going on in the human body. So the oxygen concentration in the Earth's atmosphere is between 20 and 21%. So if you're listening to me from Melbourne, Australia, it might be closer to 21% because you're at sea level. If you're in Basel in Switzerland, it might be a little bit less oxygen as you get higher altitudes, so that would be closer to 20%. But in the Earth, in the human body, as I mentioned earlier, oxygen does not diffuse very well. So the oxygen concentration in a given tissue will vary depending on how close those cells in that tissue are to oxygenated blood in the circulatory system. So in solid tissues like the liver, the heart, the kidneys, the oxygen concentration can be as low as 4%. And in the eyes and the brain, it's even lower, as you can see. And bone marrow, where the stem cells grow, has very low oxygen. And so this is why many of us culture stem cells at hypoxia. So just keep in mind that a hypoxic incubator uses nitrogen gas to decrease the oxygen concentration in the incubator, and then you add in 5% CO2 as usual. And so that can affect the function of the incubator and how your cells grow also. So I'm not going to take much time to talk about that now, just to let you know that that is another function that we can look at with regard to the incubator. Okay, so just like with temperature, I want to provide some tips to you to maintain gas concentration in whatever incubator you're using now. So the first, of course, is check your carbon dioxide calibration at least once a month using a secondary probe or a FiRite system. And check it early in the morning, first thing in the morning when you get to the laboratory before the incubator door has been open and closed throughout the day as people access their cultures. If you're using a hypoxic incubator, check the oxygen concentration first. If you want to do any calibrations, you want to manipulate the oxygen setting before then turning to the carbon dioxide. And that's because you're reducing oxygen, but you're adding in carbon dioxide. So if you increase the CO2 concentration, that can affect the oxygen measurement. Use a gas guard, an automatic tank switcher, so that if the CO2 tank runs out at night or on the weekend, you don't run out of carbon dioxide. And again, just like temperature, think about limiting the door openings and the amount of time that the incubator door is opened. If you're using hypoxia particularly, you might want to consider degassing the media, so using nitrogen to bubble through the growth media before applying it to the cells. Also, if you're working with slow-growing cells like immune cells or neural-derived cells, even if you don't have to split or passage the cells, you might want to consider changing to fresh media every few days, because as the cell number in a given culture vessel increases, so will the carbon dioxide, because the cells are giving off carbon dioxide as their metabolism, and that can exhaust the buffer in the growth media. Oh, and also use the correct carbon dioxide setting for your media. So the carbon dioxide provided by the incubator depends on the buffering capability of the growth media. So some growth media may require 8% or 10% carbon dioxide setting. So that's something to just check. Okay, so let's look at humidity. This is the last parameter of the incubator that I'm going to talk about. And it's one that's often overlooked. So let's talk about this. So the reason we provide humidity in the CO2 incubator is because it reduces evaporation from the growth media. So in very low humidity, you get higher evaporation. What happens when you get evaporation is only water leaves the culture media, and as a result, the remaining salts, minerals, hormones, nutrients, etc., become concentrated, and some of those factors can reach toxic concentrations, right? So the growth media that we use are precisely balanced for a reason. And if the nutrients or test factors, the things that we're working with to stimulate the cells, become too concentrated, that can reach toxic concentration. So it also turns out that evaporation is 4 times faster at 80% humidity than at 93% humidity or greater. Now, so high humidity is a good thing, but as you approach 100% humidity, then you get condensation. So it's essentially, 100% humidity is rain, right? So over about 95% humidity, you can see condensation in the incubator, or the water vapor collects and you see puddles or droplets of water. So that's actually a bad thing. Like many things in life, it's a balance because condensation can provide a breeding ground for microbial contamination. So you definitely want to have proper humidity to prevent evaporation from your growth media or your cultures, but also too much humidity can be bad. So it's actually a very difficult parameter to control well, humidity. So let's look at how humidity is controlled in different systems and different incubators. Just like temperature, humidity, and carbon dioxide, humidity recovery from a 30-second door opening depends on design. So this table is looking at time to recover to 90% humidity or greater following a 30-second incubator door opening. So again, left to right, the dark blue bar shows humidity provided by a protected or covered integrated water reservoir. So this is built into the floor of the incubator. And the recovery there is less than 10 minutes. That's very fast. Humidity is very difficult to recover fast. Next to that, the bar to the right, the other blue bar, is a similar built-in integrated water reservoir without a cover. And you can see that removing the cover slows recovery a little bit, and this makes sense because as you keep things covered, they're going to dry out less, so you get higher humidity. The yellow bar is an external water system. And what this is it's an external water reservoir connected to a nebulizer that creates a water vapor or mist, and that's injected directly to the incubator chamber. And that's really fast to recover as long as you have a circulating fan to speed that recovery and that circulation following a door opening and following injection of the atomized water. And you can compare that to the green bar to the right of that yellow bar, which is a similar but different system in an incubator without a circulating fan. And then the last two bars, the red and the purple, just comparing surface area of a traditional standard water pan used to provide passive humidity in an incubator. And so again, I hope I've demonstrated that different technologies produce very different conditions, even though a lot of incubators look very similar from the outside. It's the design and engineering that go into that incubator that can produce very different conditions for cells. So what can you do in your current incubator to maintain proper humidity? The first thing is keep your water reservoir full. More water equals higher humidity. But it's important to refresh that water at least 2 times per month so that you get a chance to clean out the water reservoir and eliminate any microorganisms that may have colonized that area. And then it's also important to use only sterilized distilled water because deionized or ultra-pure molecular grade water can actually cause corrosion in the incubator. Just like with temperature and carbon dioxide, if you're trying to maintain an artificial environment inside the incubator that's very different than the conditions in the surrounding laboratory, limit the door openings and duration. And this is especially important in a clean room where you may have the ambient temperature in the laboratory much lower than in the incubator. So normal room temperature for many of us is between 20 and 22 degrees Celsius. But in a clean room, if you're wearing a Tyvek suit, of course, that gets very warm, and so many clean rooms are set at 16 degrees Celsius, and they're drier than the incubator or even than a typical laboratory, which is about 50% humidity. So that increases the difference between the ambient laboratory and the incubator when you're in a clean room. Also, consider using larger culture volumes, and I assume that many of you are scaling up your culture vessels, and so of course that also helps prevent evaporation just because any evaporation that does occur will be less dramatic because it'll be dispersed through a larger volume. Ensure proper airflow. Make sure that your incubator has a circulating fan in the incubator chamber, and then that fan is properly cleaned and properly operating. And then finally, if you're using small volumes, if you're using 96 well or 384 well plates, these really small volumes are more susceptible to evaporation. And so one option there is to use this kind of unique, the Nunk Edge multi-well plates, where there's a moat surrounding the micro wells that you can fill with distilled water to help kind of cushion that evaporation and reduce evaporation from the wells inside in the center of the plate, and it also reduces edge effect quite a lot. So that's humidity. We've also talked about temperature and carbon dioxide gas. So I just want to now kind of pull it all together and show you that all of these parameters work together for cell health. So I know this is a little bit hard to read, but these are just to remind you of each of those graphs that I presented individually for temperature, carbon dioxide, and humidity. But a lot of times we don't talk about humidity, for example, and we think more about temperature than CO2, but I just want to emphasize that all of these parameters work together for cell health. And of course, it's critical for cells to spend maximal time at ideal conditions because as those conditions change, that can affect how the cells react. And then finally, I hope these graphs demonstrate that different technologies yield very different results. So I mentioned with regard to each of those parameters, temperatures, carbon dioxide, and humidity, I mentioned a probe that's in a bypass loop. So I wanted to talk about that a little bit more and describe what do I mean by that. So this slide shows my drawing of what a bypass loop might look like. It's sort of electronics for biologists, if you will. So what happens here is you can see that there's an arrow that forms a U-turn. And that represents tubing coming to and from the incubator chamber. Okay, so we're essentially in a box inside the incubator housing, but outside the incubator chamber. And so this tubing removes an air sample from the incubator chamber where the cells are, passes through a filter to restrict any airborne contaminants, passes through a pump to keep the air moving, and then encounters the sensor, which then reacts to that air sample. Now, because this is outside the incubator chamber, we also have to have a separate heater. So this compartment is also heated to the same temperature as the incubator chamber, it's 37 degrees, but it's a completely separate heater. And it's not something that you're going to calibrate if you check the temperature on your incubator. You just don't have access to that. So my point here is that a bypass sensor simply does not react to the same conditions that your cells are in the incubator chamber. It just can't. So why would somebody offer this in an incubator? And I guess the thing is that if you think about what's going on in the incubator chamber from a cell's perspective, not only, or from an electronics perspective, I guess is better, not only is that incubator chamber heated to 37 degrees, and not only is it very humid, and of course both of those things are very hard on electronics, but the CO2 gas is providing an atmosphere that is weakly acidic due to carbonic acid. And so that's very detrimental to electronics. So if you put the sensor outside of that carbon dioxide gas and outside of that high humidity, it helps preserve the life of the sensor. So in other words, inside the incubator chamber, you need very solid state, very robust components to withstand those conditions. But the important thing is that if you're using a sensor to react and to control the conditions that the cells are experiencing, then you want to have the sensor react to or experience the same conditions that the cells are experiencing, and that just can't happen with a bypass sensor. So look for an incubator with sensors in the incubator chamber that are going to experience the same conditions as your cells, don't require additional equipment in an external space. This allows those sensors and probes to constantly sample and react the conditions in the incubator chamber to react to the same conditions as yourself, and that will give you faster recovery after a door opening. Okay, now I just want to finish up talking about uniformity. So another thing that you want to think about with regard to an incubator for cell therapy, especially as you're getting to larger sized incubators and larger culture vessels, is that you want the cells, regardless of where they are in the incubator, to react to the same conditions, because the cells react to changes in temperature and pH and the concentration of nutrients in the medium. So you want the incubator to give you the same conditions regardless of where you look at in the incubator chamber. So uniformity throughout the chamber from top to bottom is important. And if I look at this a little bit closer, if you think just about temperature, hot air rises, right? So if you stay in one of those fancy hotels with a big atrium that's maybe 14 stories high, and you get off the elevator on the 14th floor, and the whole space is opened all the way down to the first floor, the ground floor, you find that the temperature is much warmer on the 14th floor in that atrium than it is on the first floor. So we know that heat rises. But that's not all. It turns out that carbon dioxide gas is heavier than air, so it sinks. And nitrogen gas used in a hypoxic incubator to decrease the oxygen concentration that nitrogen gas is lighter than air, so it rises. So without a circulating fan, you can have stratification of conditions inside an incubator such that cells at the top will experience very different conditions than cells at the bottom. And if you're doing a large production or a large experiment, if you're in R&D, you will have cells in different conditions depending on where your culture vessel is in the incubator chamber. So the only way to ensure that you have uniformity throughout the chamber is to use a circulating fan. Now, we've talked about how a circulating fan provides fast recovery, and I demonstrated that with the tables showing different engineering designs. We just talked about how a circulating fan provides uniform conditions. Sometimes you will hear that a circulating fan is bad because it dries out your cells. And that, again, depends on the engineering and design. If the fan is blowing super fast and blowing directly onto your cell cultures, yes, I can imagine it would be possible to see desiccation of your growth media and drying out of your cells. But if you have gentle airflow circulation, if it's designed to circulate quickly, but not blow directly on cultures, so that as this image shows you get turbulent, gentle airflow, then you don't see desiccation, you don't see evaporation from the growth media. And a circulating fan provides two additional benefits as well. You can provide HEPA filtration to capture airborne microorganisms that might be coming in from the laboratory, from the humans working in the laboratory. And a circulating fan reduces condensation in the incubator chamber. So condensation that would lead to microbial contamination, for example. So again, some final tips to maintain uniformity and recovery in your incubator that you currently have. Number one, of course, look for an incubator with a circulating fan. Number two, look for data provided by the manufacturer on recovery. Don't just look at specifications. So if you're comparing incubators, everybody has a list of specifications that they meet. So temperature variability, for example. But it's also important to look at data demonstrating how those specifications look. So I've got two examples here. On the left is a chart showing recovery of humidity following a 30-second door opening, and on the right is a temperature map showing a 27-point temperature map throughout the incubator chamber. Also, calibrating, of course, monthly will maintain your uniformity and your recovery. As I said earlier, keep the water pan full or the water reservoir, and as always, try to reduce the length and the number of the door openings. So let's talk about some conclusions. Just as a review, the points I want you to remember from my talk today: all incubator parameters affect cell growth. It's not just about temperature, it's not just about pH or CO2, but also humidity plays a big role in how your cells grow. And so, as I hope I've shown you, the design and quality that goes into the incubator affect the function. The location of the sensors affects how they perform, such that in-chamber sensors will experience the same conditions as your cells. A bypass sensor, which is outside the incubator chamber, simply experiences different conditions than your cells experience, and that different designs can provide different results in your process or your cell growth. So recovery after a door opening and uniformity throughout the incubator chamber, again, affect the cell growth. And so air circulation with a circulating fan is required for uniformity and fast recovery. And you can ask for these data from the manufacturers. If you think about, if you're looking at, for example, initiating a new institute, a new building, you're expanding your processes, and you're thinking about a large incubator purchase, don't just look at specifications, a list of specifications, but ask for these data, and even consider buying a single incubator from a few different manufacturers to compare how that incubator performs in your processes, because your end product depends on the cell health, and the cell health is dependent on the CO₂ incubator. Okay, so just to finish up, a few words about the nice people who sent me here. Thermo Scientific has been making CO₂ incubators for more than 50 years, and so that provides us with proven technology and engineering and performance leadership. Many of the innovations available on CO₂ incubators today were invented by our engineering team. Also, we provide factory-trained technical support and service teams. So these people go to the factory, they turn the screws, and they're trained on how these incubators work from the ground up. And they're available to help you in person or by telephone, and they know the incubators inside out. Also, because of our history, we work closely with cell culturists and cell therapy institutes around the world to try and keep up with the changing needs as we go forward in the 21st century and cell and gene therapy becomes more and more applied. We also have a lot of different choices so that we can match your incubator to your needs, and that includes everything on this list, but also we can provide customizations as needed. If you have specific design requirements, for example, you want an incubator that's built into the wall of your clean room instead of being wholly included inside the clean room, those are things we can work with you. And then contamination control. HEPA filtration, proven high-temperature sterilization that meets the standards provided by the United States and European pharmacopoeias, for example. And then we have newer technologies that have been invented in the last few years, including even better gentle airflow circulation in our newer models, newer sensor technologies. I talked about that a little bit. The water reservoir giving super fast humidity recovery, and so on. We also, I mentioned our factory-trained service teams. We can provide documents, and we can provide specialists who will come to your site to help you with IQ, OQ, and temperature mapping. And I mentioned customization. So one new thing that we have that is really designed for separating projects, and if you're a company that's working on a vaccine for COVID-19, for example, and you're designing different viral vectors for vaccine production as just one example, you might consider our cell locker system, and this is a unique design that includes 6 autoclavable polycarbonate chambers that segregate the inside of the incubator chamber into 6 individual compartments. They have membrane filters that allow air circulation for uniformity, but restrict circulation of microorganisms. And so when you open one of these chamber doors, the other 5 do not experience that door opening. So for example, if you're working with stem cells, they remain at their ideal conditions much more uniformly than if you were to open the entire incubator door. And microorganisms, as I said, are restricted. So that's all I have today. There's more information. There's a lot of technical information at our website at thermofisher.com. And Susie, I'm ready to take any questions.
Wow, thank you, Mary Kay, for that presentation. And we will now start the live Q&A session of this webinar. And a quick reminder to our audience on how to submit questions, simply type them into the drop-down box located on the far left of your presentation window, labeled Ask a Question, and click that send button. Mary Kay will answer as many questions as time permits today. And we have quite a few questions coming in already, Mary Kay. Let me start with the first one. How do you verify that your temperature is correct on the LED readout of your incubator? And there's a part 2. Can you use a thermometer placed into the incubator?
So it sounds like you answered your own question with the second question. So yes, the way that you would verify the temperature on the LED readout of the incubator is, there could be LCD too, by the way, is you use a secondary temperature probe. And you want to use one, if you're in the United States, that's NIST certified. So the National Institutes of Standards, and I can't remember what the T stands for, might be temperature. So you want a calibrated secondary thermometer, and even those can vary by 0.1 to 0.2 to up to 1 degree. So make sure you use a temperature, a secondary temperature probe that you know the calibration on, so that if your incubator says 37 degrees and your secondary temperature probe says 38 degrees, it doesn't mean the incubator is wrong. So you need to use a secondary temperature probe that has been calibrated, and what the difference, the plus or minus variation on that secondary temperature probe might be. But yes, so the idea is use a secondary temperature sensor and put that in the incubator chamber.
Wonderful, thank you. And when cells are taken out of an incubator, say for observation under a microscope, the media temperature cools down towards ambient temperature of the room. How does this drop in temperature affect the pH of the media and thus the cells?
Ooh, wow. Okay, so you're absolutely right that temperature can affect the CO2. It should not affect the buffering capability of the sodium bicarbonate or the HEPs or whatever buffer is in your growth media. So the temperature should not affect the buffering capability of the CO2, because the idea of a buffer is that it's going to take up and give off hydrogen ions to balance that pH. And certainly that's not going to change in a short term.
Thank you for that. And I again want to thank our audience for these great questions coming in. Our next question, how are vacuoles related to toxicity? Or toxicity, I'm sorry. How are vacuoles related to toxicity? I apologize. That sounds plural.
Toxicity. Related to toxicity, and that is my fault. I misspoke. I actually meant to say granules in the cytoplasm when talking about carbon dioxide and pH, not vacuoles. So thank you for calling me out on that so I could clarify that. Yeah. Granules can be an indicator of toxicity, not vacuoles.
Perfect. And what is the ideal maintenance temperature of the growth medium?
So I'm not exactly sure what you mean by that, but ideally, of course, you would store media for up to a month or so at 4 degrees Celsius, not at 37 degrees. So I hope that answers your question.
And Mary Kay, do you recommend CO2 measuring devices?
I don't have one specifically that I would recommend. Certainly Thermo Scientific does have a secondary CO2 IR sensor we can provide. There are some from other manufacturers too, and many people use a handheld Firite device. Just make sure that, just as I was talking about older style infrared sensors can drift over time, if you're using a secondary IR CO2 probe or even oxygen probe, you want to make sure that that's been calibrated in the last year also.
Thank you. And our next audience member says, we currently do a manual decon process every 6 months, but are looking at more of an automated with H2O2 decontamination or heat decontamination. What are your recommendations on routine incubator decontamination?
So my first recommendation is to look at the proof of efficacy. Okay, so hydrogen peroxide is a really great disinfectant. It's broad spectrum. It will kill pretty much anything, including the SARS-CoV-2 virus. But it's a little bit difficult to automate. So there's a lot of handling involved, and the problem with an automated system for hydrogen peroxide is that there are areas that maybe will be shadowed from the mist, and if those droplets miss an area of the incubator chamber, then any microorganisms there would obviously survive because they wouldn't be contacted by that hydrogen peroxide disinfectant. So look for proof of efficacy according to the United States and European pharmacopoeias, or the pharmacopoeia relevant for your nation. They've gotten pretty harmonized in that instead of, for example, for heat sterilization, they used to recommend a given temperature and time, so 160 degrees for 2 hours, and that would constitute sterilization. But it's now recognized that if you don't reach that given temperature, microorganisms may survive because they're not actually at 160 degrees for 2 hours, for example. So now the pharmacopoeias recommend or require proof of sterilization using 1 million organisms, resistant bacterial spores, which are proven to have a specific D value, and I won't go into what that all means right now, but my point is that sterilization is now defined as elimination of 1 million resistant microorganisms, and those microorganisms are specified biological indicators. So look for a process that has been independently tested to verify that it meets that sterilization requirement. And there are companies that will come in and do a manual chemical disinfection on your incubator, and many of those are good. Make sure that it won't corrode or cause degradation of the electronics in your incubator, and make sure that that company is going to verify neutralization of the chemical that they use and document the efficacy in the end, that they truly did sterilize your system.
Thank you for that. And again, audience members, great questions. We have time for a few more questions. Mary Clay, do you have any recommendations for how often one should change CO2 sensors? And in parentheses, she writes, Vios incubators.
Okay. Full disclosure, the Vios is my favorite incubator. So the sensor and how often you have to replace it depends on the sensor itself. So, for example, a thermal conductivity sensor is very stable and will last the life of the incubator generally. The newer infrared sensors, so if you have a Vios, it has that silicon-based infrared sensor I mentioned, and that is going to have a very long service life. Generally, the service life for a piece of laboratory equipment is expected to be 7 to 10 years. And so my recommendation is if you have an older style infrared sensor and you start to see drift over the carbon dioxide measurement, it's probably time to replace that sensor. So I guess the short answer is older infrared sensors are expected to last maybe 5 years. Thermal conductivity sensors should last the life of your incubator, especially if you're keeping the calibration and the humidity constant. And then the silicon-based infrared sensors should approach the life of your incubator lifetime.
Thank you for that. And here's our next question. It's 2 parts. What kind of device do you recommend to use to measure CO2 level? And if the CO2 level was wrong, is it easy to modify CO2 levels by ourselves?
Depending on the incubator, there should be instructions in the user manual for modifying the calibration, or basically what's the offset of the CO2 sensor. For replacement of the sensor, I would recommend a factory-trained service specialist. And I hope I answered the question. If I didn't quite answer that correctly, let me know.
Rankia, how do you control variable oxygen concentrations if growing a variety of cells at a particular time in the incubator?
So so an incubator has one oxygen sensor generally. If you're going to use different oxygen concentrations, you would need multiple oxygen sensors. So a single incubator can control oxygen to one particular setting, whether that's 1% oxygen or 5%, so on. We do offer a small modular chamber that holds 12, I think it's 12 Petri dishes and 9 multi-well plates. I might have those numbers a little bit off. And that can be placed inside any incubator to control oxygen, so you could test with multiple of these small chambers. You could test different oxygen concentrations. The thing to know about that is that it does require some external equipment in order to provide that oxygen. So you need a canister of premixed gas with the concentrations of gas that you're interested in testing. You need a pump, you need tubing, and you need a regulator. But once you get all that set up, you just put your cell vessels in the chamber, you charge the chamber with gas, and then put it in the incubator, and it's a good way to test multiple oxygen concentrations simultaneously.
Thank you, Mary Kay. We have time for one more question, and hopefully you can help this audience member. She says, in my lab, since a few days of amniotic cells do not grow after the first initiation, but after first media transfer to second T25 flask, the cells start to grow, and the growth after next media addition is very unpredictable. What's going on? What's wrong with this?
So I haven't grown amniotic cells myself, but the oxygen concentration in the uterus is very low, usually between 2 and 5% oxygen. And when you have primary cells, such as cells, I'm assuming these are primary cells, yeah, they're kind of maybe in shock, you could say, even if you're simulating the oxygen concentration in the uterus. So if you're not simulating the oxygen concentration, then that can play a role here. So for most primary cells, I would recommend a hypoxic incubator. So if you look at a graph of primary cell growth in an incubator that is controlling oxygen to simulate the in vivo conditions and one that's not, in the one that's not, you might see the cells grow for a day or two when first removed from the body, and then as you describe, you'll see a lag where they won't necessarily die, but they'll just kind of be in stasis for a little while. They're cellular couch potatoes. And then after several more days, you might see the growth curve start to go up where they're actually starting to multiply. What's happening during that lag is that the cells are actually mutating genetically to adjust to those artificial conditions. So I would encourage you to look at hypoxia and do some research in the primary literature to see how other people are incubating those cells and what conditions they're using, because that can play a big role in how primary cells grow, especially if you're looking for them to mimic conditions in the body. You want them to be genetically similar to, the way they were in the body before you put them in culture. So I hope that helps.
Wow, thank you very much, Mary Kay. And thank you for this great presentation today. Do you have any closing remarks for our audience before we close today?
I guess I would just say, to reemphasize, a lot of the information that I presented today was unknown to me when I worked in the lab. I kind of assumed that an incubator is very similar to another incubator, and I hope I've demonstrated that in practice that's actually not true, and that can have effects on your cells. So if you're thinking about a new incubator purchase, especially if you're in a cellular or gene therapy company, and you're thinking about a large purchase, one thing to do is to consider buying a single unit for you to test, to run through the IQ, OQ qualifications that you might be looking at, because that's going to affect your processes in the long run. And that's it. Thanks, everybody, for attending. I really appreciate it. And thanks again to Susie and everyone at LabRoots for a great format.
Mary, thank you for your important research. Before we go, I want to thank the audience for joining us today and for their interesting questions. And as a reminder, any questions that weren't answered today will be addressed by the speaker via the contact information you provided at the time of registration. I'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share this email with your colleagues who may have missed today's live event. Until next time, stay safe, be healthy, and have a great day. Bye-bye.